Good afternoon, everyone, and a warm welcome to the Twist-sponsored session held at JCA AACR 2021. Twist is very happy to participate as a sponsor in the first JCA AACR, and we are very excited to see so many of you join us today. My name is Mark, and I head the sales division in the Asia Pacific region. In this Twist-sponsored session, I am proud to be joined by our Senior Manager of NGS Solutions, Brian Hogland. He will share more about Twist's industry-leading target enrichment solutions that combine quick customization and content flexibility with unprecedented sequence efficiency. You will also hear from his presentation data that illustrates target enrichment efficiency through diverse sample qualities using Twist's complete solution. The data will demonstrate how Twist enables researchers to sequence more deeply and more efficiently into each sample. Before we start, let me share a brief introduction about Brian. Brian earned his Bachelor of Science degree in Cell and Molecular Biology from the University of California, Santa Barbara. He spent the next 16 years at Roche working as a research scientist on the in vitro diagnostic assays, lab-developed tests, and NGS in oncology, transplantation diagnostics, infectious disease, and companion diagnostics. In 2017, he transitioned to the role of field application scientist with the Roche NGS team, and then later that year brought those skills to Twist. As a Twist field application scientist, he supported customers working with our Twist portfolio of products for NGS. Brian now holds the role of Senior Manager in the NGS Business Solutions, where he has combined his expertise in the field of NGS with his passion for helping customers develop novel, useful, and cutting-edge products that accelerate today's NGS-based research. Without further ado, may I invite Brian to kick off the presentation.
Thank you, Mark, for the introduction. Let me share my screen and get set up. Perfect, thank you. So I would love to be here in person, but unfortunately we still cannot attend in person. So I hope I can reach you all at a good level. I'd like to spend a little bit of time of first introducing Twist and what we do here at Twist. So Twist was founded in 2013. 5 years later, we did have our IPO. We started and headquartered in San Francisco. We've moved our operations into South San Francisco with a business and technical team in San Diego. And we've been expanding throughout the globe, Tel Aviv, Singapore, and Guangzhou. So what does Twist do? Quite simply, Twist makes DNA and we make it very well. So we have a multitude of different product lines. In our synthetic biology portfolio, we have genes, oligo pools, and libraries. Today, we'll be focused on our NGS platform, but also we have a data storage program and a biopharma program as well. So the Twist NGS target enrichment benefits, we focus on three key things. Number one is maximized efficiency, followed by greater flexibility, and of course, fast panel optimization. It all starts with the power of our platform. So we have a platform that enables oligos to be synthesized in a scale that's unprecedented. Approximately 1 million oligos per writer run has a scale and speed that is really unmatched in the marketplace. To combine with the power of our platform in making DNA, when we moved into NGS, we focused heavily on our design capabilities to design custom panels as well as fixed content panels. So we took this process through a rapid design, build, test, learn cycle of a machine learning. So quite simply, we would design a panel, we would synthesize it, test it, and learn from it. The things that we would learn from that would be thermodynamics, GC content, repetitive elements within the target space. We would learn on how those thermodynamics affect hybridization capture and build that back into the design to learn how to boost the appropriate probes at specific concentrations to then create the most optimized panel performance possible. So what is this kind of maximum efficiency that we speak about? Quite simply, it's get the most amount of information from the least amount of sequencing. So the diagram listed below here is kind of a standard base call pile-up cartoon. So in the dashed lines is your sequencing depth. Your sequencing depth is important for you to establish what is the correct depth to achieve a marker or a base call accurately. So we want to focus on the useful data. Anything that is above that line is actually over-sequenced and precious real estate is taken up on the sequencer. Anything below the line is under-sequenced. That under-sequencing cannot give a determination of your base call and is unusable. Lastly, there is the off-target. When a hybridization probe is in the solution, you'd want to bind it to your target and not binding elsewhere, creating unusable data. Next is providing the flexibility. So we weave in the flexibility into all components of our system. So starting here with the workflow. In the green is where we fit in the central dogma of the sequencing space, from sample prep, library construction, target enrichment, sequencing, and then tertiary analytics. So we built out a multitude of ancillary reagents that are able to penetrate different applications and ultimately markets to give the end user the tools that they need to seek the answers that they desire. Next is fast panel optimization. So we have a pipeline of operations and design to get you your design of your custom or your fixed panel content at a very fast pace. Usually takes about 3 weeks from panel design to synthesis to shipping out. In this process, we do have the ability to do a very small kind of panel for you in terms of reactions to where you can test that, pilot that. We can look at that empirically, perfect that panel for you to then launch it through your production and large scale ups of your project. Now I'd like to talk a little bit about the product that we have that has been available for approximately a year and a half, and that's the Twist Comprehensive Exome. So as you all know, exomes are highly utilized in disease space as it makes up approximately 80% of the disease targets that you look for. So this comprehensive exome is appropriately named in the fact that it's the most comprehensive content across all of the relevant databases that are out there. So when we compare to competitors, when we look at the RefSeq, CCDS, and GenCode, we're over 99%. Content is very important, but also you have to have performance. So to the graph on the left, what we have is the target enrichment percent bases covered. So at 20 or 30x, when we compare it to our competitors, we achieve a 95% coverage at a specific sequencing depth of all the panels. And that allows you to, again, have confidence in your base calls. Now you can take this exome and through our other ancillary reagents, you can incorporate a Covaris instrument for adaptive focused acoustics that allows for work with FFPE samples, very challenging samples. So utilizing a protocol that we have in collaboration with the company, we have taken this component and you can do a focused acoustics, library construction, a hybridization capture of the exome sequencing, and then get your results. So before I show data, I'd like to talk to you a little bit about FFPE. As you know, this particular sample input is highly variable. So what a lot of researchers do is they have to determine first the quality of that FFPE that they're entertaining and see if it's even viable for this type of sequencing. So there are a multitude of assays that are on the market, one of which is a qPCR assay. qPCR assays are really to see how many amplifiable molecules are available for this. And what has been determined in previous literature is that with Q ratios, where you take basically one primer, one forward primer, and 3 reverse primers, and you amplify to see the size, that ratio will then determine the quality of the material. So it's been deemed that a quality of 0.2 is actually sequenceable. Anything that falls to the left of that, the quality is too poor to sequence. But based on the performance that we've been able to perfect in our hybridization chemistry, our design, the quality of our oligos, we're able to recover that poor quality FFPE samples. So we can take this benchmark of 0.2 Q ratio, and we can actually push this over all the way down to about 0.05. So that's why we tend to say we sequence the unsequenceable. We're able to gather all of the benefits that we have in our entire system and pull those answers out that you need. Now I'd like to change course a little bit and talk about methylation sequencing. This has been a project ongoing at Twist for approximately a year and a half that I've been able to be a part of. And we see a lot of utility in this. So that's why we refer to it as a new era in epigenetics. So we use our hybridization capture system to get the same exceptional performance, the unparalleled uniformity, sensitivity, that rapid panel design iteration, flexibility, and it's available at a wide, broad range of panel sizes. So whether it's 50 kb all the way up to a size of a methylome of 85 megabases, we have exceptional performance to get the answers with the correct screening tool that you need. So in this strategy, we had to take an approach where, again, using the power of our platform, we can synthesize oligos at an unprecedented scale. So we had an advantage to where we could take and make probes for the unmethylated and methylated species of that, the sense and antisense. So what we actually do in this methylation strategy is we create probes for all 8 different flavors of what would occur after you have an enzymatic conversion of the methylation, where every probe will be more or less designed specifically for that methylation state. And that's what gives us this unparalleled sensitivity that we've been seeing with our end users. Another important thing is that we want to create an end-to-end workflow. Creating panels is just one stop of this, but really building that full, complete end-to-end solution is really what we try to achieve. So at Twist, we make DNA, we don't make enzymes. So we have to reach out to our partners that are experts in that field. So we went out and we worked with a company called NEB. They had a new library prep technology called NEBNext EM-Seg. We licensed it in for Twist targeted methylation sequencing. This is a different approach. This is a enzymatic conversion of your methylated cytosines, of your unmethylated cytosines. It is not your standard traditional bisulfite, and there's a lot of distinct advantages in that. Throughout the rest, we've created a fast hybridization system to be able to get your sample to sequencer within a day in the target enrichment space, and also a Twist methylation enhancer that is a blocking mechanism that achieves even higher specificity. And then, of course, most importantly, the Twist methylation custom panels. We're able to design those very rapidly and turn them around to you. So a quick illustration of the NEBNext EM-Seg. It is different in the sense that it will still get you your hydroxymethyl and methyl groups, but what it does, instead of using bisulfite, it uses a mixture of enzymes and workflow. The first one is a TET2, which is a protective. So it puts a protective block on the methylation signatures. You then have an oxidation enhancer. You denature that. And then the ApoBec enzyme comes in and deaminates the cytosine, turns it into uracil, which in later stages in PCR is converted to an adenine or thymine. So this enzymatic method, the first thing you must have to assess is the conversion rate. You have to have a very high conversion rate. So the graph diagram on the left illustrates this, where the enzymatic conversion rate is approximately over 99.7% to the gold standard traditional bisulfite of 99.6%. If you look at the chart on the right, this is the yield after PCR. So when all things are controlled, a standard out-of-the-box bisulfite control will yield you approximately 10 nanograms per microliter. Through extensive workflow optimizations, we were able to achieve a larger yield, but the enzymatic method right out of the box greatly established the highest yield overall. Now, this is important in oncology, cell-free DNA. So the cell-free DNA patterns have a very traditional multi-peak pattern. The bisulfite method degrades this very high level molecular weight that is indicative of the second and third histone turn modifications. The enzymatic method maintains those as there is important information that can be contained in that. So taking this through this library prep, putting it on a capture. This is a specific panel that we ran for early validation studies. And in this specific panel, when you look at just controlling for enzymatic versus bisulfite, you see a great improvement in your depth of coverage, your uniformity, your specificity, and your library complexity, which is illustrated as a duplication rate here. So this is a panel that I'd like to discuss. This is one of the early collaborators that I worked with in building out this application. It was a researcher in Cambridge, UK, and they have been working with a panel for approximately 2 years with cell-free DNA for the application of early cancer detection. They were working with a different vendor for approximately 2 years, 4 to 5 panel design iterations. They were willing to test our utility, our solution, and right out of the box, it had great performance. So the fold enrichment was approximately 3 times higher. The uniformity, where lower is better, was significantly better. So all regions sequenced more or less equally. The 2 different specificity metrics of on target and off bait were greatly elevated in the twist system. And this was, again, right out of the box from the design. So those are performance metrics. So what does that really mean in terms of actual utility for your application? Well, when you're looking at early detection cancer screening, you're mainly detecting the DMRs, the differentially methylated regions in your tumor versus normal sample. So with the regions of interest here, there was a cohort run of 4 tumor, 4 normal, where we ran the experiment and the methylation patterns were called out. The goal is to draw a very clear, distinct line that differentiates tumor versus normal to where you can actually see that sequence and that species in the population is in fact cancerous. So now I'd like to announce the new panel that we have that is launching this month. It is the Twist Alliance Pan-Cancer Methylation Panel. So as the name says, it is a pan-cancer. We have 31 cancer types and 47 different disease entities that are broken down. Please reach out to our Twist affiliates and sales team to see if these specific oncology related diseases are contrived in this panel. It's approximately 1.5 megabases, 13,000 probes. There's 126,000 CPG sites involved. And all these CPG sites are then delegated across promoters, 5', 3' UTRs, exonic, intronic, and intergenic sequences. So I'd like to walk you through a proof of principle study. So using the end-to-end Twist methylation system workflow, we have the NEB library preparation kit, followed by the target enrichment system and this panel. So a cancer sample cohort of 30 samples total, 15 pairs of tumor versus normal were run in this, 3 different types. So there was 10 samples of lung tissue samples that were from the FFPE format, so 5 malignant, 5 benign. And then there were additional 20 samples that were fresh frozen. That was 5 breast cancer of malignant and benign, and then 5 colorectal cancer of malignant and adjacent tissues. The end result was sequenced on the NovaSeq 6,150 end of base pairs, and the following few slides are the results. So the hybrid capture performance metrics. This is something that I have to look to analyze to look at the performance of the panel to see if we can ultimately get to your end goal. So the first box here is the uniformity metric of the fold 80 base penalty. Lower is better where 1 is perfect. So to achieve a fold 80 of approximately 1.45 is really unprecedented in hybridization capture technology, especially with methylation. The way that I have this broken out is broken out into different sequencing depths. I'll get to that point on the next slide, but different sequencing depths ultimately determines your sequencing economics and how much power you have to put into it. So now moving over to the next one is a specificity metric. In this term, it is 1 minus % off bait. So across all different sequencing depths, it achieves approximately 83.5% sequencing specificity. The next most important thing is the library size. So the library size are the unique molecules that are actually captured, critical when you're looking at very low inputs. So again, here, through the different sequencing depths, we are able to fully saturate out all of those unique molecules. Now, the inverse of library size and unique molecules is the % duplications. So the % duplication rate is a function of that sequencing depth. So as you look at the more you sequence, the more duplicates are sequenced along with it. And you come into a system where the more you sequence, the less information you get out of it. So sequencing depth is kind of critical. So here is kind of the sequencing depth and coverage metrics. So the first one is the pass filtered reads of each system when it's downsampled to its specific raw coverage. The mean target coverage, which usually is what people tend to most look at, and the % target bases, the percentage of target bases seen 30 times. So looking through this, we can kind of put our hands on the fact that we would like about 250x raw coverage, which equates to about 3.5 million paired end reads, which means you can approximately put 225 samples on a NovaSeq run at high output. Now, some of the other specific methylation metrics. So the first things we have to look at, again, is that conversion rate. The sample has to be converted. Those unmethylated cytosines do have to be deaminated, and we need to track that. So in looking at this in split from benign, malignant, and normal, what we can see here is that we have a very high 98% of the very hard fresh frozen and FFPE samples, where the normal is approximately over 99.5%. Now over here is the average per sample across the entire 1.5 megabase space of the called CpG ratio. So you can see again in a different graphical visual where you have an elevated hypermethylated signal in the malignant versus the benign and normal samples. And this is something you want to track. Now, again, this is in the full aggregate over the entire panel. So we want to look at that at specific markers. So this is a heat map that was filtered by a t-test. The 3 different tissue types are broken out below here with the samples of each one illustrated here. Just very visually striking, you can clearly see that there is a strong difference in the elevated methylation when it's relative to its counterpart. And you can tell that from the heat map. So you can see which markers are spotted out in breast cancer, colon, and lung. When we take a different statistical approach, the DSS test, it's a dispersion shrinkage sequencing of differentially methylated signatures. So here, just taking a different approach, you can see the difference in the markers on what you're looking at. And again, I like this because it's extremely visual for you to illustrate. Another thing, now this is interesting in kind of when you're looking into the application. So this is hypermethylated regions of the transcriptional start sites. So in the middle, you see a hypermethylation of this breast cancer. And then as you go upstream of KB or downstream of KB, the methylation patterns tend to slow down. So it's a very interesting finding. You clearly see what's going on in a malignant versus a benign tissue sample. This is another way to illustrate it, where we look at a specific gene in the panel, we pull it out, TDR-D10, it's involved in breast cancer. And what this shows is a very hypermethylation of the promoters, and this results in a downregulation of gene function. So the top are the normal samples, where the below ones are the tumor samples and the malignancies. And you can clearly see an elevated pattern of methylation that can then help researchers determine what their end question is to be. So in summary, we would like to say that the results of this, it really demonstrates the unique capabilities of our system that we've built out. We highlight the excellent performance across a wide range of methylation targets and levels with a highly optimized out-of-the-box design and the flexibility to be able to choose among those different levels of design stringencies. So featuring efficient and gentle enzymatic conversion, the high uniformity methylation capture, these are new tools to push the envelope in the development of highly sensitive epigenetic applications. So thank you very much for your attention. I really appreciate it. Please reach out to Twist for any further information, and we'd love to meet you to talk with you about your project and your goals. Thank you.
Thanks very much, Brian. Much appreciated for the presentation today. I have one quick question. Could you name some of the companies that are working with Twist in their NGS solutions?
Sure. So what we will have to say, we'll have to go with the ones that are publicly available. There is a group that I've been working with for quite some time that has come out and published information with a product launch of Helio. Another group that we have been published, that we have in a publication that is peer-reviewed literature, is Grail, where they use our oligo probes. Past that, there are many more, but they are not public at this time. So we'll have to leave it to their discretion for their publications to show.
Understood. All right. Well, look, thanks everyone for joining today. Thank you, Brian, for staying up late and presenting. And we wish you all the best. Thank you.
Perfect. Thank you very much.